Okay. Yes. Okay. So the general outline of my talk is I'll discuss what protein design is and why people are interested in it. I'll discuss computationally how we've modeled protein design using a framework of probabilistic graphical models and searching for low-energy amino acid sequences. And then I'll discuss the main topic, which is the design of promiscuity or multiple functionality into protein sequences, and discuss some of the trade-offs that are inherent in such design. So again, what is protein design? So the goal of protein design is to engineer proteins to have some specific biological function that you're interested in. But since to a large degree, it's believed that structure determines function, so overall the computational protein design problem is typically reduced to search for a particular structure, target structure, and you'd like to find an amino acid sequence that will fold to that structure. And this is also known as the inverse protein problem. So in protein folding, you start with a sequence and you want to predict the three-dimensional. So why do people do protein design? It started off, I guess you could say, with people being interested in modeling the biochemical properties of natural proteins for stability, etc. And recently there's been a lot of work on designing novel proteins or novel structures or novel functionalities, enzymes, structural proteins. And what we're most interested in is actually using protein design as a tool to gain a view of the protein structure and protein sequence space and see how well protein energetics and protein structures can be modeled and what we can learn from that. So when people perform computational protein design, one of the assumptions that they often make is you start off with a particular protein structure, and in this work we're going to assume a protein structure with a fixed backbone. And in addition, we're going to model the amino acid side chains in a discretized manner. So I'll describe what that means in a second. So basically, each amino acid side chain can have up to 4 dihedral angles. So we're going to model those dihedral angles. Instead of naturally, they could assume continuous degrees of freedom. So here we're going to model them using discretized versions of those, using what's known as a rotamer library, which is basically a statistical analysis of side chains culled from the PDB, the protein data bank. Okay. Now, computationally, one of the problems, even with using this simplified model of protein of amino acids, there's still an exponential number of possible conformations, even for a particular amino acid sequence. And again, we're trying to model protein design, so you actually have an exponential number of amino acid sequences as well. So computationally, there's a large search space to go through when you want to design a protein structure. So that's one of the problems. So I'm going to talk about our search algorithm in a minute, but in terms of the modeling, we have to score each amino acid sequence and each conformation. So people have come up with all sorts of protein energy functions. Okay, so basically these energy functions consist of 2 parts. One is usually some sort of physical atomic energy interactions or function, van der Waals forces, electrostatic interactions, salvation energies. And in addition, people have culled from the PDB, from the protein data bank again, statistical or empirical potentials and combine those into modern day energy function, which are some of the well-known ones and some of the ones I've used in the research that I'm going to show here, include EGAD, Rosetta, Orbit. But one of their main features is that in order to be computationally relatively simple, is that they use a pairwise decomposition of the conformational energy. That is, only pairs of atoms are considered, not higher order, like let's say triplets or whatever. And that you end up with that, like the energy of a particular conformation R, that basically is rotamer side chains, is at most the sum of pairs of rotamers. So again, that's to simplify things here, but again, it's still an NP-hard computational problem. So what we've done, first I'm going to talk about, okay, so to recap, computational protein design, you start with a fixed backbone protein structure. You decide which positions you might want to design based on the surface or in the particular problem. Then you model the amino acid side chains using rotamer library. You calculate energies using your particular energy function that you're interested in using. And again, now the goal is to find low energy conformations and low energy amino acid sequences with those low energy conformations. And the question is, again, how you do that, because we're dealing with an exponential search space, and like I said, it's an NP-hard problem. So what we've done is used a probabilistic graphical modeling framework. So I'll give an example here. So let's say you have this structure to design, and you have this toy example of 4 residues that you're designing for positions. So you build this graphical model of these 4 positions. You connect all interacting positions with an edge, and each edge contains a pairwise energy matrix where each of the matrix contains interaction energies between the side chain at a particular amino acid at one position and a particular amino acid, or a particular rotamer at the second position. And now you want to use this graphical model to predict the lowest energy sequences. So what we've done is we've used a Loopy belief propagation algorithm, which is a well-known algorithm in the field of probabilistic graphical modeling. And I'll go over that quickly. So basically, what happens during the Loopy belief propagation algorithm is that messages are passed from neighboring nodes, from one neighboring node to the other neighboring node. In this example, node 3 will be passing messages to node 2. And the contents of these messages are basically probability vectors, where there's a probability for each rotamer of each amino acid type. And the way that these messages are calculated is using the input messages from neighboring nodes itself. So in this case, nodes 1 and 4 will send input to node 3, and node 3 integrates that input with its direct interactions with node 2, and you end up with the messages that it is its preferences that it believes node 2 should assume, which rotamers and which amino acids. And this process continues iteratively until hopefully the algorithm converges, and at the end, each node, which is each residue in the protein that you're designing, will know the probability of each amino acid and each of its rotamers. So in the end, what you can do is you can choose the highest probability rotamer, and you end up with the lowest energy amino acid sequence. And we've come up with some algorithms that generalize this and allow you to predict multiple low energy sequences, and I'm not going to go into that here. So basically, what we've shown is that we have an efficient framework, and we've shown that it works quickly and finds very low energy sequences compared to some competing algorithms using this belief propagation framework. So now the main goal of this research again was to design protein sequences that have multiple functions, or in this case, you could view it as having multiple structures. So one possibility would just be to design for one particular structure, you get sequence 1. You could design only accounting for the second structure, you get sequence 2. But of course, the problem would then be that sequence 1 in structure 2 will have not such good energy. Sequence 2 in structure 1 won't have good energy either. What you want to do is design one sequence that will be capable of folding to both structure 1 and structure 2. In this case, it would mean the same. You'd have one sequence that can bind both protein, actually peptides in this case. So using our graphical model framework, what we've done is, in this case, again, let's say you have these 2 structures. So what we've done is, if you formulate the graphical model for each of those 2 structures, as I described previously, using again the nodes and the interaction energies between them, and then what you do is you combine those 2 graphical models, one for each structure, into a single graphical model. Interactions between the graphical model ensure that the corresponding positions will choose the same amino acids, so that you end up with a single sequence that is compatible with both structures, in this case, structure alpha and beta. So just as a sort of sanity check, what we saw was that here we were using the transducin beta protein. So we designed the interface of transducin beta with 3 different binding partners. And what we saw was that, at least energetically, so at a computational level, is that when we design for each particular, we optimize the transducin beta binding to each particular partner, we get low energy for binding that partner and high energy for binding the other 2 partners. And that was true for each of the 3 partners that we optimized for in our design procedure. And then if we did that for each pair of partners, again, we find sequences that are compatible with binding that particular pair, but not the third one, and that was true for all pairs. And then what's important was that when we actually optimized for binding all three binding partners, we were able to get single sequences, or in this case, actually 100 sequences, that each sequence is capable of binding each of the three partners. So that was nice, but that's just sort of a computational result. What we want to know is what happens to the biological quality of these sequences as we add additional partners. So to do that, again, we were predicting 100 lowest energy amino acid sequences compatible with binding in every subset of partners, 1, 2, or 3 partners. And what we used is the JSD, the Jensen-Shannon divergence, to compare between the profiles that we predicted and, let's say, evolutionary profiles, in this case from the ISCB database. So overall, what we did see is that as additional states are added, as additional partners are modeled, then we end up with sequences that are more similar to evolutionary sequences. That is, they have a lower JSD score. So on top here, you see the ISCB profile on the left. One-state designs, those are the designs of the transducin beta interface that bind one particular partner. And these are the transducin beta interface designed to bind two particular partners, and these are those designed to designed to bind all three partners. And overall, there was a slight trend of decreasing JSD, so we were happy with that. So that was our question was, can we show this in a more statistically robust, large data set? Sorry. Okay, so again, we wanted to focus on these promiscuous protein binders, let's say the ones that are the hubs in protein-protein interaction networks, and it's well known that they evolve more slowly due to the multiple constraints that are applied to them. And one of the important things to note here is that they face opposing forces during evolution, since on the one hand, you want them to bind multiple partners. On the other hand, each partner should be bound with reasonable affinity, otherwise it won't actually be considered binding that partner. So our question is, how have these sequences evolved to favorable interactions with so many partners? So what we did was we took calmodulin protein, CAM, which is known as a calcium in the cell, and it's known to bind hundreds of targets in the cell, and we culled from the PDB database 16 high-resolution calmodulin bound to peptide targets, all of which bind in the 16 structures here. And what we did was we determined the common interface, which was 20 positions in total, that all 20 positions interact with all the partners, or at least 3/4 of the partners. And this is the binary profile for these 20 interface positions. And now our goal here was, again, we wanted to predict 16 sequences or 16 possibilities where each of these sequences would be optimized for binding just a single partner, one of those 16 binding partners. We would do 120 calculations which were optimized for binding each pair of calmodulin interactors, 560 calculations of triplets of interactions, and we did one calculation of sequences that are optimized for binding all 16 structures that we were dealing with. And actually for each such scenario, we designed 100 lowest energy sequences to come up with these profiles. So these are just some examples of the profiles. I mean, the details aren't important here, but these are the profiles for one state that is one partner, two partners, three partners, and on the bottom, all 16 partners. So I'm going to discuss this now. So one of the first things that we asked was, can we reproduce the phenomenon that we saw earlier about the addition of partners? Does that yield more biological or more natural sequences? And we saw that it does. Just on the right side, what you see is that the number of mutations compared to the wild-type sequence decreases from 9.5 to 7 to 6 to 4 as additional partners are modeled. This is on average, of course, and you see that this is the distribution over here. So we were happy with that result, and now we wanted to go into details about some of those results. So what we saw is that there are, on a position-by-position analysis, there are some positions, such as this position, that are conserved. That is, for each, these are four, in this case, four different possible binding partners. And we saw that no matter which partner you choose, the calmodulin design comes up with methionine in all four scenarios. On the other hand, there's this type of position where each partner induces a different sequence at the calmodulin interface. So we wanted to sort of quantify these different positions and categorize them. So what we did was we used, again, we used the JSD score to calculate the 16 partners, and we defined those positions to be those that are highly conserved between structures, and the specific positions to be those that are diverse between the different structures. That is, each partner induces specific requirements on the Kalamazoo interim. And one of the interesting things that we observed was that the affinity determining positions, which were categorized again by high conservation between the different partners, actually have quite strong intramolecular energies. These are low energies in the wild type structure, or wild type sequence actually. And vice versa. The specificity determining positions actually have quite strong intermolecular energies. So basically we're able to conclude that it seems that the intramolecular interactions are critical for the binding affinity, and they basically stabilize calmodulin in this binding mode, whereas the intermolecular interactions are critical for conferring specificity to any particular target. But again, since these targets have different requirements for impose different requirements on calmodulin, the question is how are those requirements sort of integrated to obtain sequences that are compatible with binding two targets at a time, let's say. So that's our question over here. So, I mean, one possibility is, again, what we saw before, is that both targets, let's say, one target, this is another target, this is the design for two targets simultaneously. So in this case, you see that both of them impose methionine and calmodulin and together they impose methionine. So that seems almost trivial, although it's not obvious that that would happen. The similar case, now a more interesting case is where one target imposes lysine and methionine, another target methionine, and together though they require methionine or this target wanted. So this target's sort of energetic needs are common in this case. So that was an interesting scenario. A different scenario is possible when one target might prefer one amino acid, one another, together they come up with a third amino acid. So that was also a different possibility. Overall, what we did see is that very often the two targets already, if they didn't agree, then the most common was this preferred one scenario, which is this one over here, where one target's preference were chosen over the other. That's sort of like a statistic. The question is, in those cases where the two targets didn't agree, how did that affect the sequence quality, evolutionary sequences? So what we saw is the most common scenario where one target imposes one amino acid, but together they impose something different, and that's actually beneficial as compared to the evolution. This beneficial position-by-position analysis shows that is often the case, and that explains how the number of mutations compared to wild type goes down as additional states are incorporated in the modeling. About that. So one last thing about the energetic compromise, that are required when integrating these multiple constraints at the energetic level. So again, we have, let's say, 100 sequences designed for a particular structure or a particular partner A, 100 for B, 100 for C, 100 that are compatible with binding partner A and B, 100 for B and C, and 100 that are compatible with binding A, B, and C. So the question is, what happens to the energies as those constraints are integrated? So what we see here is a histogram of decrease in stability in binding of partner A. You have a calmodulin interface that already binds partner A to additionally bind in a second partner, partner B. So you see that there's a decrease in stability of up to 10 to 15%. On the other hand, once you already have a calmodulin interface that binds both A and B, two particular proteins, and you're required to bind the third protein, partner C, then the decrease in stability is much less. It's maybe up to 5% or much smaller usually. So our conclusion from this is that it seems to be, at least for the calmodulin analysis that we did here, that the biggest hurdle is going from binding one partner to binding more than one partner. And that's indicated by the larger decrease in stability here versus here. And we saw some similar results when we analyzed the jump from binding one partner A to binding additional partners, A, B, and C, where, again, you see that the big jump is going from 1 to more than 1. Okay, so just to conclude, so overall, what we saw was both with transducing beta and with Kalmogylin sequences, that when you design for interaction with additional natural targets, you actually get sequences that are more similar to native sequences, and that's similar to some results that were observed previously. And what's important is, of course, that there is some degree of trade-off that's necessary, but we observe that the main compromise is in going from binding one partner to binding more than one partner, and after that, it seems to be less of a compromise necessary. And that's it. Thank you. I want to thank my advisor, PhD advisor, Michal Lineal, my collaborators on this project, Julie Schiffman and Chen Yanover, and you, the audience, and for funding, for ISMB funding. And just one brief note is our open source software for performing multi-state protein design is now available at this link. Thank you.
Thank you very much. Time for questions. Yeah.
I have a question regarding conformational flexibility and fit. Kalmogylin so happens to undergo a dramatic conformational change.
Yeah.
So that leads naturally to the question of induced fit in protein binding.
Okay, so since the structure of Kalmogylin that we modeled was already down to each of those partners, the induced fit was already, I guess implicitly, or it was already modeled in the way Kalmogylin was. Okay. So it was already modeled in the way that Kalmogylin was. It might be more of a problem if we were modeling Kalmogylin both unbound and bound, but since we're dealing with Kalmogylin bound in the same binding mode for all the partners, then it's less of a problem. But you're right, it's definitely an issue.
Miki?
Yeah, you exactly asked one of my questions. The other one is, can you tell us a bit more about your energy function that you use for the scoring?
Yes, okay. So, I mean, it was part of two different projects. So the main energy function that we used for the Kalmogylin design was the orbit energy function. What do you want to know about the energy function?
You gave an example of semi-physical energy function. Is that what you used? Or
yes, okay. Basically, the orbit energy function is in that category. It's actually a even more physical energy function than some of the others, but yeah. It has van der Waals term, under Jones potential, that is. What else does it have? Electrostatics. I mean, those are the main components in the tight bonding.
Okay, if there are no more questions, then let's thank the speaker again.